I'm sure about now you have probably noticed the sudden increase in oil prices recently. It sure does take a toll on our pockets, huh?
Well, let me tell you a little something we call biofuel.
What is it, you may ask?
Well, it may be something that can actually save us from this crisis. I am Dexter Dan Anthony G. Camano.
And I am Shaw Bimoto.
And I am Beatrice Giobalde.
And we are researchers from STEM of Galianosa National High School. And we are here to discuss to you our study, which is entitled Guava Leaves Powder Extract as an Alternative Source of Bioethanol. One of the most widely used energy today is fuel. Fuel provides energy services such as heating, transportation, and electrical generation. Because of its many uses, it is a critical component of the global economy. It is highly demanded in every country, and international corporations are highly involved in importing and exporting fuels. However, due to the Russia and Ukraine crisis, global oil supply forecasts have significantly changed into today's global oil prices.
Bioethanol fuel has a vital role in the field of environmental conservation. By mitigating global warming and conserving fossil fuel, it is mainly produced by yeast fermentation from different feedstock that mainly contain fermentable sugars or carbohydrates.
Ethanol is a highly flammable liquid similar to gasoline. It is produced and burned, which results in carbon dioxide emissions, a greenhouse gas. However, ethanol manufactured from biomass is considered carbon neutral in the atmosphere.
Guava, with the scientific name of Psidium gajava, is a small tropical tree or shrub. It is grown in tropical and subtropical areas worldwide and cultivated for its edible fruits.
Looking into this perspective, the researchers evaluated a potential source of bioethanol from guava leaves. The researchers explored the production of bioethanol from guava leaves because of its 48.13% carbohydrates, hemicellulose including 7.71% xylose, 22.6% arabinose, 0.59% mannose, and 29.41% galactose, 21.2% lignin, and 40% cellulose, hence making it a potential source for the production of bioethanol.
The researchers aim to produce bioethanol by utilizing the lignocellulosic contents of guava leaves. Specifically, the researchers sought to answer if there's a significant difference between the efficiency of varying amounts of guava leaf extract in producing bioethanol and if there's any significant difference between the alternative bioethanol and commercial fuel in terms of flammability and duration of flame.
The majority of the studies that were aforecited are focused upon the use of various biomasses for the production of biofuel, mainly for bioethanol, which according to Zabed and his co-researchers on 2017, bioethanol can generally be derived from sugars, starch, and lignocellulosic biomass. Different types of biomass were utilized to improve or create alternative sources of biofuel.
For the purpose of the study, quantitative research design will be utilized, particularly through experimental research design, since the subject will be gathered randomly and will be tested in different variations.
The researcher will be conducting the experiment while utilizing approximately 300 grams of guava leaves. The method for which to gather the guava leaves will be random sampling and will be tested indiscriminately. The commercial fuel will then be labeled as the control group, and the varying amounts of extract from 50, 100, and 150 grams of guava leaves will be the experimental group of this experiment. Production of bioethanol will undergo several processes which will be thoroughly explained by the researchers. Phase 1. Harvesting. The researchers will firstly collect guava leaves from its tree.
Phase 2. Pre-treatment or grinding. The guava leaves will be washed thoroughly and will be air-dried for 7 days. Then, will be grounded into powder through the use of blender and will be set aside into 3 containers.
The next phase will be hydrolysis or extraction. The hydrolysis process will act as an extraction process for fermentable sugars from guava leaves. Phase 4 will be filtration.
This process will be used in the study to filter the extracted fermentable sugars from the guava leaves powder.
Phase 5 is heating. The filtered fermentable sugars will be subjected to 70 degrees Celsius heat through the use of digital hot plate to increase the rate of hydrolysis.
For the next phase, fermentation, this process will be utilized for obtaining ethanol by adding 1.5 grams of yeast, sealing the concentrations with cheesecloth, and setting this aside for 4 days.
Phase 7, distillation. This distillation process will be used to extract the ethanol from the fermented sugar and water.
Phase 8, dehydration. This process will be performed to remove the excess water contained in the ethanol.
After the 8 phases, the obtained ethanol from the varying amounts of guava leaves powder will then be subjected to flammability test, both from the control group, which is the commercial fuel, and the experimental group, which is the bioethanol produced from guava leaves.
The researchers will utilize the one-way analysis of variance because there will be a comparison of varying amounts of bioethanol being tested. It will be applied to determine whether or not there are significant difference between the means of the commercial fuel and bioethanol made from varying amounts of powdered guava leaf extract in terms of flammability and duration of flames. Therefore, using the said statistical tool will provide the study a valid, reliable, and justifiable result.
We would like to continue and develop our study for the times to come.
And we hope that our product will soon be utilized by the local citizens and our community.
Thank you for listening, and that is all there is to our humble presentation.